Hi, everyone. Welcome to Foresight's Biotech and Health Extension seminar series sponsored by 100+ Capital. Thanks a lot for joining, everyone. We're starting relatively rapidly this year with a bunch of new seminars, and this one is one that people have been looking forward for quite a while and has been in the making for a little bit. So thanks a lot for coming on today. I do want to take the opportunity also to remind folks that we do have our workshop coming up in this series. And so if you're interested in meeting other folks here in person in this group or across other groups, there's a bunch of other workshops too. I really welcome you to apply to our Biotech and Health Extension Longevity Frontiers workshop. So we have a bunch of people already confirmed that are also in this group. And we have like the format and everything already on here. But last year, it was, I think, a really good time. The workshop is on our website, and I'm also going to share it again here in the chat for those of you who are in here. But applications are open now, and it's going to be in April. And usually it's a nice way for people in this group or like folks that aren't in this group yet to get to know each other. But for now, I'm super excited and delighted to have Joao Pedro de Magalhaes here today. I hope I didn't totally butcher your name. Thank you so much for joining us. We've certainly been tracking and following your work for quite some time at Foresight. And yeah, you're now really a really wonderful, I think, scientist that has been like doing quite groundbreaking work and like pretty ambitious work and has been really outspoken about this work in the longevity, like larger space. And today you're really like going all out there with a scientific quest for immortality. And so the stage is yours. Please go ahead, take away your presentation. I'm going to be in the chat monitoring your guys' questions here, and then we're going to start with a Q&A. So thanks a ton for joining. It's been long overdue, and we're really excited to see your presentation.
Well, thank you very much. Well, thank you, Alison. It's a pleasure. I mean, I've been I started attending the meetings from the group. I suppose during the pandemic, and it was fantastic, so to have other people to listen to talks and to have other scientific discussions. So it's a pleasure to be here. As I said, I've been lurking in the shadows for quite some time. So I let me share my screen. Give me 1 second. Yeah, so what I prepared was really an overview. I mean, I'll start a little bit about what are my goals, what I want to do, and then I'll briefly summarize what I'm excited, or some of the projects I'm excited about in aging research, in the biology of aging, and then I'll talk a little bit about cryopreservation as well, and essentially how to achieve my childhood goals. So that's the idea, and I think I have like 30 slides, so it should take about half an hour, and then I'm happy to discuss. I think that's kind of the purpose of today is this to be like a two-way conversation, not just me talking. So so let me start going back in time a little bit. So basically to when I was a child, because I think I'm a little unusual in the regards that I decided what I wanted to do with my life, with my career, when I was probably quite young, probably 7 or 8 or 9 years old. So that's a photo of me about that time. And essentially, that's when I realized that everybody dies, and that not just everybody dies, but it's quite inevitable because we all age. And so it's inevitable that we all going to age, and my parents, my loved ones would die one day, would age and die, and so would I. And that really, I never came to terms with it. I think some people make their peace with death. Some people try to pretend it never happens, or try to. I'm unashamedly terrified of death, and I've always been. And I say unashamedly because I think there's a human tendency to things we cannot control. We just try to cope with them. I mean, obviously, religion would be the most obvious. I never cope with it. And so that moment I decided, okay, well, aging is going to kill me and kill everybody I love, then I'm going to find a cure for aging. That's what I'm going to do in my life. So quite an early age, this was, more than 30 years ago, I decided what I wanted to do in my life and what I wanted to do in my career. And so I studied Studied life sciences. I studied microbiology and cell biology, and many years later, I have my own lab. So I now lead the genomics of aging and rejuvenation lab. So I was in Belgium, I was in Boston at Harvard, and then I had my lab in Liverpool for quite a long time, for 13 years. And then I moved to the University of Birmingham, where I'm a professor, and I lead the genomics of aging and rejuvenation lab. So in a way, you could say that I'm trying to achieve my childhood dreams. I'm trying to find a cure for aging. That's what I wanted to do as a child. And in a sense, I guess I'm trying to pursue that. And I mean, we do quite a lot of different things. I don't have time to go into details about all the research that we do, from evolution. We do a lot of cell work as well, various computational approaches. I'll briefly mention the computational approaches and some of the work on longevity, drugs that we've been doing as well. We've also done a lot of work in long-lived species like the naked mole rat, the bowhead whale, monkeys, and so on. So we've done a lot of evolutionary work that I'm actually not going to mention today. We also develop various databases and tools and methods that I'll very briefly only touch some of the methods. So what I thought I would do, okay, so what am I excited about? Well, there's a couple of things I would like to make a contribution. I think a lot about, when I retire, what have I achieved, or what I may regret I didn't achieve. So I try to address that. I try to think of projects that excite me, that something that I'll be proud of. Not just publishing papers and getting grants, but trying to do something that will have an impact. So there's a couple of things that I'm excited, which I'll very briefly go through. The first one is that we don't really understand aging. I would say that aging is still an unsolved problem of biology, like I think Medawar has an essay on this from the 50s. So we do not know why we age, in the sense of understanding what are the mechanisms, what are the cellular molecular mechanisms driving aging. There's a lot of theories, as I'm sure this group knows, and it's been discussed here. There's a lot of theories on cellular and molecular mechanisms of aging, but they could be wrong. We really do not know for sure if they're right or wrong. There's very little empirical evidence, even in mammals, particularly for humans. There's very little empirical evidence that these mechanisms are indeed causative of aging. And as I'm sure you're familiar, or many of you are familiar with the hallmarks of aging, this collection of hypotheses. They're not known facts. I think sometimes people take them too seriously because we don't know if they're right or not. We don't know if they're true or not. So they're hypotheses. They're not proven yet. I mean, they might turn out to be true. They might not. They all might turn out to be misguided. So that's, I think, a possibility. So we don't understand the process of aging yet. And actually, last year, actually, it was 2 years ago. We're already in 2023. Me and David Jams in London, we published this critique of the hallmarks of aging, essentially arguing, said that it's a wonderful review, it's a wonderful introduction to the field, but please don't take it too seriously. And maybe there are mechanisms of aging as well beyond just molecular damage, which is the assumption of the hallmarks. And one hypothesis that we're interested in, myself and David, is that perhaps aging is also due, I wouldn't say damage is not relevant at all, but I would say aging perhaps is also due to programmatic mechanisms set during development. So processes that causes changes during development, changes that occur during development, whose trajectories are set in adulthood and continue in adulthood and become detrimental. So that's something that I'm quite interested that, for example, I think it fits quite well with all of the work of Steve Horvath and others in finding that you see this epigenetic change extremely well conserved from birth. Actually, it's not just from birth, it's from nearly conception, days after conception, all the way to old age. And I think that's quite remarkable. And it tells us something important about the biology of aging and how it may be linked to developmental mechanisms via epigenetic changes. So anyway, so that's something that I'm interested in, in trying to understand aging, or trying to develop a model that explains aging. I mean, I'm working on a couple of articles in this topic at the moment. So it's something I'm interested in. And I think, okay, what do I want to achieve in the next 10, 20 years? Developing models that explain aging, that is one of trying to understand why we age. That's one of the things that I'm interested in. Now, but of course, okay, that doesn't really solve the problem. of that I defined in my childhood, which is that we're all going to age and die. So we need to develop interventions for aging. And so that's something I'm quite interested in, and we've been doing a little bit of work on that, which I'll, again, very briefly go through, just some recent work we've done. So I guess I'll start with just taking a step back. So one of the things that we've done for quite a long time is developing computational methods for various, particularly for aging and longevity, but also for age-related diseases, done it for cancer and so on. I mean, I've always been a computer geek, so I think it's about following, doing what you love. I again, back to my childhood, I've always loved computers. I love programming computers. I love statistics. I know that's kind of a strange thing to say, but I love statistics. I do enjoy doing the statistics from the experiments or from the work that other people do in the lab, something I quite enjoy. So we do a lot of development of methods. And although we do experiments as well, we do a lot of machine learning, we do a lot of network analysis, we do a lot of computational bioinformatics analysis for various applications. But the only thing I'll focus today is more in terms of identifying new targets and identifying new drugs, identifying new compounds, particularly in the context of aging and longevity. So again, it can be applied for many different contexts, and I'll come back to this when I talk about cryopreservation. But in the context of what I'll just briefly go through, it's in aging and longevity. So, I mean, to give one just recent example, we've done quite a few of these methods, but we did this work with Alex Freitas in the University of Kent, Computer Sciences. So we basically developed this method for predicting new longevity drugs based on machine learning. So what we do is we look at existing drugs that impact on longevity, for which we have a database called DrugAge. And then we look at the targets, we look at the chemical properties, we look at which phenotypes it's associated, look at which genes that are known to be associated with aging already. So basically, we do this data crunching. We take all the information we can get from these drugs and their association with longevity and aging, and then we essentially try to predict other drugs that have similar features, similar properties. So that's essentially how it works. Obviously, there's a lot of statistics behind it, but essentially that's how it works. And so there's a preprint on this that we've just posted, well, last month, I think. So that's kind of one example of how we can use these methods. And again, they can be applied to other phenotypes as well, to other processes. So then one of the things that we did, or one of the things that we then tested some years ago already, was we developed this, again, computational method. We call it the network pharmacology method for drug repositioning. So basically, we take gene expression data from longevity manipulation. So we actually use caloric restriction from cells, but basically we take these gene expression signatures from longevity, and then we take gene expression signatures from drugs, which are available on public databases. And then we try to identify drugs that induce similar or overlapping gene expression signatures to caloric restriction or to longevity. I mean, we published this some years back already. I mean, basically, we then tested. We actually tested this in C. elegans in worms. We started doing experiments in worms, which is something that was new for our lab. We tested in worms, and 5 drugs we tested, 4 of which extended lifespan. One of them was rapamycin, which I'm sure you're aware extends lifespan in worms, in mice, in flies, which we use as a sort of positive control. But we identified several new compounds as well. One of them was a compound called allantoin. Now, allantoin is basically a marker of oxidative stress, but it's also used in skin creams, anti-aging skin creams, actually. So it was quite interesting. And I mean, these results have been validated by other labs in Singapore in particular, that allantoin extends lifespan in worms. Then we're interested in the mechanisms and interested, okay, can we repurpose allantoin? So allantoin is quite safe, but what seems to happen is that it's degraded by the digestive system. So it's not really something you can take orally. So if we wanted to do experiments in mice, or if you want to potentially apply to humans, have to be via injections, which is not really practical. So we started looking for other compounds that may work in a similar fashion to allantoin. And I mean, to make a very long story short, and this was the work of well, various collaborators, and Dominic Bennett was a PhD student in our lab. So this was basically work of several years in the making. I think it started in 2017 or 2016. So this was 6 or 7 years of work that I'll just briefly summarize in a couple of slides. So basically, we decided to focus on a compound called rilmanidine. Now, rilmanidine, first of all, it was also a compound that we found in this analysis. So in our bioinformatics analysis, rilmanidine was also there, not as top as rapamycin or allantoin, but it was also there in the compounds we predicted to be a longevity modulator. So it was a hit from our previous analysis, and there's a lot of advantages to it. So we think it works by targeting these particular receptors called imidazolin receptors, which possibly, or we Dominic thinks that's also potentially how allantoin works. So there is some overlap in terms of mechanism. At least that's how we think. But then we focus on rilmanidine, and we haven't done much work on allantoin. But the big advantage of rilmanidine is that it's an oral drug. So it's an antihypertensive drug. So it's given to patients already. So people, including elderly patients, they take it. There are side effects, but they tend to be rare and non-severe. So it struck us as the kind of drug that, if you're thinking about a drug to reposition for aging, that might work. And so we decided to focus on it for various analyses, which again, I'll briefly summarize. So the paper is now in Press in Aging Cell. I mean, it's actually available online already. So you can check it out. But basically, we show that rilmanidine extends lifespan in worms, which is what you see here on the left side. So this is various doses of rilmanidine. This is survival by age of worms that live only a few weeks. And what you see is a dose-dependent response. So up until about 200 micromolar, it extends lifespan, and then 300 and 400, it doesn't extend as much. So we focus on 200 micromolar from this point on. We show that it extends lifespan when fed to early in life or late in life. We think it works via autophagy upregulation, which I'm not going to show here. It's not synergistic with caloric restriction, so we think it works via as a caloric restriction mimetic. So we did various experiments in worms in collaboration with Colin Hewald and also Vadim Gladyshev. We showed, or we observed, that rilmanidine in mice induces similar transcriptional changes to other life-extending interventions, like caloric restriction. So again, we think it's working via as a caloric restriction mimetic. That's I think we have some data for it. So I think that's a fair assumption, although it's not 100% sure. And so we also know that rilmanidine is already used in successfully in mouse models of neurodegenerative diseases. So there's already been studies by David Rubenstein, actually, who's been on the Foresight group, giving a talk that rilmanidine has benefits in some models of neurodegenerative diseases. And so it's, I would argue it's a promising new longevity drug, a promising new geroprotector. That we identified. Now, there's not a lot of patients. I've actually tried I work in a hospital, and I try to talk to people who work with hypertension, and they said rilmanidine is not normally what you use first, so there's not that many patients, at least in the UK, that take rilmanidine, and that's been one of the limitations in trying to, because obviously we like to look retrospectively whether patients that take rilmanidine have some health benefits compared to other antihypertensive drugs, a bit like was done for metformin. But unfortunately, at least in our cohorts that I've spoken to, there's not enough patients taking rilmanidine. But that's something I'd be very interesting to explore further now, and also do further mouse studies in normal mice, not just mouse models of disease, but see if rilmanidine in normal mice extend lifespan. So that's something that I'm very interested in doing next. So, the take-home message is quite a promising new geroprotector that may have applications that they may be repurposed for other indications. So, okay, but rilmantadine, even in worms, extends lifespan up to, I think, nearly 20, but I think it's 19 point something percent, if I remember correctly. So that's not really curing aging. And, if we're talking about curing aging, we're not going to do it with drugs. Or I'd say it's very, very unlikely we can do it with a pharmacological perspective. So, so if we want to achieve the dream, the goal that I had more than 30 years ago when I was a child of curing aging, I think drugs I wouldn't say it's impossible, but I think it's unlikely, or this type of longevity in drugs, at least it seems unlikely. So we're doing another project, which again, I've very briefly summarized, which is focused on cell reprogramming. For those not familiar, so you can reprogram cells with Yamanaka factors, and you turn them into pluripotent cells, and this can turn an old cell into a young cell. So you can use partial reprogramming, which is essentially reprogramming for a shorter amount of time. Well, that's an oversimplification, okay? But it's essentially a milder form of reprogramming that doesn't change it doesn't induce pluripotency. So the cells preserve their identity, but they're still rejuvenated. So I'm sure many of you are familiar with this, and so it's something that we're exploring as well. I think scientifically I'm quite interested, or academically I'm quite interested in the topic from an academic perspective, in the sense of, is this true cellular rejuvenation? I mean, what are the potential mechanisms? How is this achieved? How can you rejuvenate a cell? Is there some selection going on? So I think academically, I think is a very interesting topic. And I think in terms of applications, they could have applications as well. So that's something we're exploring as well, specifically with a company called Youth Biotherapeutics, of which I'm CSO. And our goal is to develop gene therapies using partial reprogramming. So that's our goal. Now, obviously, there's a lot of caveats still in terms of partial reprogramming in applications. We don't know. There's very limited data in vivo still. I mean, there's a preprint showing it extends lifespan modestly in mice. But so there's still not much. There's a lot of data from cells showing that it can rejuvenate cells, according to the biomarkers we use now. But whether it works in vivo and whether it will work in the clinic, I mean, that's still a lot of question marks on that. So, but I think it has a lot of promise. And when we think about, okay, what's the most promising therapy for actually, reversing aging, rejuvenating human beings, I would say reprogramming is the most promising one. Doesn't mean it's going to work, but it is the most promising one. So that's another line of work that we're focusing on. Now, okay, so that's very promising, but what really are the prospects that we can cure aging? And so many of you, I'm sure, are familiar with this, the Stockdale paradox from James Stockdale. So James Stockdale was a pilot during the Vietnam War. This is him next to the airplane. And he was shot down over North Vietnam. He was captured. And he was a prisoner of war for quite a long time, I think 7.5 years, if I remember correctly, during which time he was tortured, beaten. I think both of his legs was broken, numerous injury. So and one of the things he noticed while he was a prisoner of war was that there were quite a few POWs that they were very optimistic, that they felt, okay, this is hell, horrible, but hey, American is going to win the war, we'll be home by Christmas. And those over-optimistic individuals generally didn't make it. So he came up with what's called the Stockdale paradox, which is essentially that you need to keep faith. You need to be optimistic. You need to believe that you will prevail in the end. So you need to always have the optimism that you're going to win. In the end, you're going to survive, or you're going to get out of that situation. But you also need to have the ability, what calls the discipline, to confront the brutal facts of your situation, no matter how bad they are. And I think this is a good analogy for, when I think about my childhood dreams and what I wanted to achieve in my career, but where are we now? I mean, that is something that is important, because yes, there's been progress, but when you think about how that's progress going to pan out in the next 20, 30, 50 years, what are our prospects? And so when you think about medical advances and aging, I mean, there are some brutal facts and some harsh realities to keep in mind. I mean, one of them is that there's still a lot of unsolved medical problems in areas that have a lot more funding, a lot more conceptual understanding, a lot more models than aging. Obvious example is cancer. I mean, we understand cancer to I would say a greater degree than aging. There's thousands of scientists working on cancer. Well over $200 billion have been spent on cancer since the war on cancer was declared in 1971. So definitely there's a lot of research, and yet there's still many types of cancer that don't have a cure. Some of them, like pancreatic cancer, for instance, there's really not significant advances in terms of therapies from 50 years ago. You think about Alzheimer's disease. I mean, Alzheimer's disease receives over $2 billion per year from the NIH. I mean, that's a calico every year that goes to Alzheimer's disease. We don't have any therapies for Alzheimer's, any substantial therapies for Alzheimer's disease, type 2 diabetes. Now, you can say, okay, but those are complicated diseases. Those are complex. I mean, we don't understand Alzheimer's disease. We don't understand what causes Alzheimer's disease, cancers, there's many types. It's very complicated. Okay, well, we don't have a cure for chronically dry skin or dandruff. There's no cure for dandruff. Dandruff, we understand what it is. Okay, maybe we don't understand everything about it, but we understand it better than Alzheimer's. There's no cure for a dandruff. So what it means is that it's actually, you know, there's a non-infectious diseases in particular. It's really hard to develop therapies for them. And I think aging has particular challenges as well. So I mean, at the moment, there's no intervention that has been proven to delay human aging. I mean, there's a lot of promising evidence from animal models. Yes, there's a lot of longevity drugs. I think our drug age database is like 2,000 compounds. I mean, none of them has been proven in human beings. The most we can extend lifespan in rodents is about 50%, which is caloric restriction, which was discovered in the 1930s, so nearly 100 years ago. So we haven't really progressed all that much in this regard. When you think about longevity drugs, even rapamycin extends lifespan about up to 15%. Never know for sure. I know there's males or females. One of them it extends more. I always mix them up. So that's in mice. I mean, mice, of course, you can cure cancer in mice all the time. It doesn't mean that that's going to work in humans. And as I'm sure you're aware, there's a lot of hurdles towards validation. You know, as drug development pipeline has to go through various trials, which are expensive and time-consuming. So there's a lot of problems. So I mean, I did this review perspective 9 years ago in rejuvenation research on whether aging could be cured in my lifetime, and I argued no, we will not, or it's very unlikely will we cure aging in our lifetime. And I still stand by that. I don't think that anything has changed in the last 10 years. You know, what I've said before about caloric restriction, that still holds true. That's still the most we can extend lifespan in mice. So of course, you know, if we're not gonna cure aging in my lifetime, so what's the alternative? And that's where cryopreservation comes in. So I mean, initially put the term cryonics, but cryonics is kind of, well, it has a reputational issues, let's call it that. So that's where biostasis come in. And then the choice between biostasis, of course, and there's been talks in this group before on biostasis, between eternal oblivion or a potential future life. Now, I'm not convinced that cryonics will work in the sense of allowing someone to be revived in the future. I think the chances are quite low, but you know, it's non-zero. And hence, it gives a possibility of future revival, even if the probabilities are low. And so I've been focusing increasingly more on cryobiology. I mean, we've did very briefly a little bit of work with Greg Fahey that this group is familiar with as well. A few years back on gene expression, looking at molecular signatures of cryoprotectant and trying to study cryoprotectant toxicity. Last year, 2 years ago, we're in 2023. 2 years ago, we published this quite comprehensive review with Roman Bauer, Jose Cordeiro, and others on cryopreservation, what we know, what we don't know about cryopreservation, essentially trying to set a roadmap for, what are the next steps, what sort of research we need, what are the gaps in the knowledge and the gaps in the technology to advance cryopreservation. The review focus on cryopreservation in general, not just biostasis. So, so I'm quite interested in advancing cryobiology in general. I don't think we're going to cure aging in my lifetime. I think it's very unlikely we will cure aging in the next 100 years. So, and I'm happy to discuss it further if anyone's interested. And so I'm interested in advancing cryobiology because I also am not convinced of the current protocols being used in cryonics. I think there's a lot of problems, a lot of damage that's being generated. So, so we need to advance the state of the art in the field, and that's something I'm quite interested in doing. And as I said, I'm trying to increase research into cryobiology and cryopreservation research. And one of the advantages of cryobiology is there's no need for clinical trials. I mean, okay, depending on the applications, there'll be regulatory steps, but the clinical trials, excuse me, in the framework of the FDA that I showed you, that's not really necessary. So it's easier to develop. Excuse me. So it's easier to develop applications in cryobiology than in aging or in cancer. So there may be low-hanging fruits that will allow us to advance the field and have broader applications, not just in cryonics, but in cryobiology in general. So we've been trying so one of the things I'm trying to push now is the application of our methods, the methods that we've successfully used so far to identify new genes, to identify new drugs, in particular at the pharmacological level, trying to apply this in silico, this computational machine learning methods to cryobiology. So essentially trying to develop new compounds or new formulations that are safer, they're more effective, that minimize toxicity and optimize cell survival. So that's something that I'm doing. And so one of the things I'm doing together with Roman Bauer that I mentioned already, and also Emil, who was presented in this group not too long ago, Emil from Tomorrow Biostasis is starting a company to do this application of these technologies, ultimately to improve formulations, which could have commercial applications in cryobiology in general. But really, my motivator has never been about making money. It's always been about living forever, I would say. And so the goal is to try to develop the technology to improve formulations for biostasis. That's the ultimate goal. Although, as I said, there could be a lot of other applications. And so we're developing this company called Oxford Cryotechnology. I don't live very far from Oxford. I live between Birmingham and Oxford, actually closer to Oxford than to Birmingham. And so we're starting this company in Oxford called Oxford Cryotechnology, exactly to advance research and technologies and applications in cryopreservation. And so with that, I think that's pretty much my time. So I'll just summarize what I've told you so far. So as I said at the beginning, I think death is really, really, really bad. I mean, people are aware it's bad, but I think there's still this notion that, oh, I'm not afraid of dying. I mean, I've seen a lot of people tell me that, which I do think it's almost like a psychological coping mechanism. But so I think it's important that we acknowledge that it's really bad and that maybe we can do something about it. Maybe there is an alternative. Maybe life can have a happy ending. And of course, aging is the first or the major obstacle. And that is what we need to overcome. So ideally, we would find a cure for aging. Although, as I said, I think that's going to be difficult. One of the reasons being that aging remains a mystery of biology. We still don't understand why we age. I mean, there's a lot of theories, DNA damage, epigenetics, and so on, but we still don't understand why we age. Although that's one of the things I would like to investigate or try to develop new models of why we age in my academic research. I've also told you of our studies of longevity drugs, in particular allantoin and rilmanidine, which is a new longevity drug. that we hope to, well, we think it has potential for repurposing. Whether it will work for other diseases, I don't know, but I think there is potential for repurposing rilmanidine for other age-related conditions, maybe even for slowing down aging. I told you of our work on, mentioned briefly our work on cellular rejuvenation therapies with Youth Bio, which I think is really the most promising for developing rejuvenation technologies, and I think it could have a lot of applications. I think there's a lot of potential to it, although, I mean, whether we could cure aging, I think that's I think that's perhaps over optimistic. And I mean, as I said, I do think aging is unlikely to be cured in our lifetimes when you see the pace of progress that not just in the field of aging, but also in other medical areas. And that leaves us to cryopreservation and biostasis. And I'm not persuaded of the current protocols, and that's why we need to improve cryopreservation protocols. And that's something that I want to focus on, and we're trying to develop this company in where we apply machine learning, computational methods broadly defined to cryobiology to improve formulations that may have broader applications. And so with that, well, so this is actually I was going to say the new office. I moved here last year. So I work at Queen Elizabeth Hospital in Birmingham. So actually my office is here on the left. At University of Birmingham. And so if you're interested, I mean, this was a very broad overview. If you're interested in reprints or more information about our work, check out our website. Feel free to drop me an email if any of this is of interest. Of course, this is our lot of talented and ambitious, skilled students and postdocs. And of course, thanks to our funders as well. And thank you all for your time and attention.
Awesome. Thank you so much. This was really great. And you're clearly doing quite a bit on that front. Okay, wonderful. Well, you're making your younger self proud. I think that's always a nice thing to do. Okay, we have a first question here from Micah. I also have a bunch, and it's just a reminder for people here in the chat to either drop their questions in the chat or raise your hand. Both work on my end. Okay, Micah, you go first.
So you mentioned that you're pessimistic about achieving significant longevity in your lifetime. I'm curious if the delivery and timing problem with cellular reprogramming was solved. Does that significantly change your prediction, or do you think that cellular reprogramming, even if we could deliver it everywhere and time it perfectly, still wouldn't address the problem?
I think there's other potential problems to reprogramming. The obvious one is cancer. I mean, I mean, some of the earlier work from Manuel Serrano in doing reprogramming, this was not partial reprogramming, this was full reprogramming in mice. Basically, the mice developed tumors. So there's still a lot of question marks whether, it can be done safely, particularly if you're talking about multiple tissues. I think if you're talking about a very specific tissue, I mean, for example, the work of David Sinclair in the eyes of mice, okay, so that there was a relatively short-term reprogramming, partial reprogramming, actually variant technique, and that that worked. But when you're talking about doing it in a more systemic fashion, my other worry, in addition to the technical issue that you mentioned of how to do it technologically, even if that could be overcome, then my next worry would be cancer. You may end up with a lot of tumors. So that's the other problem we need to solve. And I mean, on that context, I mean, one of the things we're doing with Youth Bio, exactly trying to develop safer combinations of factors or even novel factors that induce cellular rejuvenation without increasing the chance of developing cancer. So that's what we and I'm sure many others would like to do, but it's still not solved yet. So there's other problems besides the delivery, or other challenges that need to be overcome.
Okay, wonderful. We have John next.
Hi, Joao Pedro. Good to see you again.
You too, John. How are you?
I'm doing well. I'm in Florida. I have a couple of questions about A lanolin, you said it gets digested in the digestive system. Could it be taken intranasally or sublingually?
That's a good question. Short answer is I don't know. I don't know. I don't know if anyone has explored it even. Maybe. I mean, I think that's a good suggestion. I don't know. I'll try to look it up. I'm not aware of any work along those lines, but maybe. I mean, there's a lot of applications of allantoin, particularly in skin creams, as I mentioned. So maybe someone has looked into it. I know in mice it has to be, or at least what's been done so far is with injections. There's been some experiment in rodents already, and that was with injections. But yes, or I think that's a very good suggestion. Thank you, John, and I'll check it out.
If I were to self-experiment with it, what sort of dosages should I consider?
I wouldn't use. I wouldn't suggest you self-experiment with allantoin. I think. Well, the problem is if you take it orally, it'll just get digested. So it doesn't.
No, I mean sublingually or intranasally.
I don't know. I mean, drop me an email. I mean, there are certainly dosages from rodents that have been used already in terms of injections. So probably you would be able to extrapolate from that. But again, I'm not recommending it.
What about real manatine dosages?
So real manatine does has dosages. I don't remember off the top of my head, but there are established human dosages for real manatine. Having said that, I mean, it's still a prescription medicine. As I said, it's used to treat hypertension. But there is established dosages for real manatine. So you'd be able to find it out. Maybe it's even on, you'd be able to find it out. Or as I said, I don't remember it off the top of my head. But if you drop me an email, I can point you in the right direction.
Okay, I'll do that. Thanks very much.
Thank you.
Awesome. Anton, if you can unmute, you're next.
Yeah, hello. So I actually
Anton.
wanted to ask, do you hear me? I'm a bit noisy
Yes.
place.
Yes.
Yeah, so I actually wanted to ask 2 questions. So first, I'm a bit upset that you're so pessimistic after doing so much research, but don't you think that there is a promise that adjacent technologies, I mean, we have huge progress in machine learning, in genetic engineering, in bioprinting, that they can actually speed up biomedical progress and especially aging research a lot, because they will allow testing more stuff in parallel and with better methods. And the second thing is that I didn't quite got from your presentation. You said that you're now doing some cryopreservation research, but I mean, maybe I missed it, but you didn't say what 4 problems we should focus in terms of cryopreservation that are either crucial for cryonics or that look like low-hanging fruits that we should solve as fast as possible to get nice applications. Thank you.
Thanks, Alton. Yeah, nice to see you again as well. So let me ask. I'll start with the second question, maybe. So in terms of the cryopreservation, one of the things we've started focusing a few years ago, or I say tried because it's been difficult to get funding, but also talent, to get students interested in cryopreservation. So it's been something that's been quite slowly. Or it's not as fast as I wanted. It's not progressing as fast as I wanted. So in terms of the question, it's cryoprotectant toxicity. So that's been one of the major, because you need to use for vitrification in particular, you need to use very high concentrations of cryoprotectants, which of course increases toxicity. So that's something that we've been focusing is the cryoprotectant toxicity. And so what the goal for Oxford Cryotechnology is exactly to tackle that, to try on one hand to develop formulations that increase cell survival while minimizing toxicity. I mean, that can be done in more than one way. I mean, if we could discover new cryoprotectants, that would be one way. If we could optimize concentrations and the combinations of existing cryoprotectants, that's another way. If we could develop or identify or repurpose cryoprotectant toxicity neutralizers, that would be another way. And that's what we want to explore. So it's addressing the cryoprotectant toxicity problem, which is one of the major roadblocks in cryopreservation. When you go from cells to organs, I mean, I'm not even talking about human beings, whole human beings, but even in organs, that's already becomes, in larger organs, that becomes a problem already. So that's one of the big roadblocks we want to address. So that's your second question. Your first question was about what about existing technologies? So that's a question I actually get a lot, which is that, I mean, there are a lot of promising technologies. I mean, I am optimistic about the potential, for example, for machine learning. I mean, we're doing machine learning to optimize or to prioritize, I think would be a better word, better prioritize drugs for longevity or for aging or for other applications. That's what we're doing. And absolutely, they can help. I do think they can catalyze faster progress. The question is whether the progress is going to be fast enough, because I mean, if you look at it, we know of a lot of longevity drugs, for instance, already, but they still don't do better than caloric restriction in mice. I mean, that's the problem. Yes, we know of more drugs than we did 10 years ago. It's a very rapid increase. If you look at, I mean, I published a couple of recent papers on this, but it's in terms of the effect sizes, it's still not good enough. So that's the problem. So the question is whether we can identify drugs that actually retard aging further or more than caloric restriction. And that's a big question mark, I would say. So finding more drugs and using machine learning, AI, all of these technologies to find more drugs, yeah, absolutely. But are the drugs going to have a bigger effect than caloric restriction? I am not so convinced about it. And that is the big problem, is whether we can find drugs or we can find interventions, not just drugs, but interventions that retard aging more than caloric restriction. That remains the gold standard in the field. And even caloric restriction has its problems, as I'm sure you're aware. It doesn't work in some strains of mice. So even in mice, it doesn't always work. So that's the problem. And the last point I would make is that, of course, we still have to go through validation. So even if you have something very promising in aging, you still need to do a validation. In mice, that's going to take you 4 years. In human beings, a clinical trial will take you several years and cost dozens, if not hundreds of millions of dollars. So even if you have some interventions and therapy that's very promising, it still needs to take time. And that's not something that technology can easily overcome. You can get more candidates, but you still have to go through these steps. So that's one of the big problems I think we have in aging. That, for example, in cryobiology, it's much more streamlined because you don't have to go through these long clinical trials. I mean, even in cancer, because cancer, you don't need to do a 4-year study in mice. Even in cancer, it's easier to test drugs and compounds and so on than in aging. So that's, I mean, I hope that answers your question.
Well, I was just thinking about this organ-on-chip models that can have some promise to iterate faster. I know now they're pretty far from being good models, but if bioprinting to get like larger organs, gene editing, and organ-on-chips can become larger, it means it can speed up at least cellular and mouse works. Because if we look into cell culturing, it's kind of haven't changed for 17 years, and this actually flowed a lot.
Mm-hmm. I agree. I think those are very promising. Absolutely. I mean, I think there is. I don't want to come across. I'm quite optimistic about technology in general. I think there's a lot of things we know about genetics now that we didn't know 10 years ago or 20 years ago. So there has been progress, but turning that basic science progress into a clinical application. I mean, that is the big difficulty, and that's, I mean, you could question the framework of the FDA and so on, and that's something. There's been some interesting discussions here in the Foresight group as well, but it's still quite a complicated, expensive, and time-consuming. That's one of the big roadblocks.
Okay, thanks for answering.
Thanks, Anton.
Next one up, we have Ageless Partners.
Hey, good morning. Thank you. Really appreciate everything you're doing. Very well said across the board. Just had a question. I mean, in terms of timeline on some of these therapeutics that you're looking into, and kind of your perspective on the approach that Brian Johnson, who was on the Foresight call last week, you know, when I understand his approach, you know, you're kind of getting this immediate feedback right away. You can measure and quantify biological age across all your organs, 70 plus organs. And you could actually see rather immediately whether a therapeutic works. So I was just curious on your perspective on that. What are kind of maybe the trade-offs? I'm sure there's some error on the estimates of biological age across those organs, but it still seems like a promising approach given you kind of get almost immediate feedback. So I'd be curious on your perspective on that approach. Thanks.
So thanks. Okay, so I think personally, I'm not very adventurous. That's why I try new things. I will exercise. I do exercise like you're doing now, and that's something I do regularly. But you know, I'm not the kind of guy who installs the latest version of Windows on the first day. You know, I wait for other people to do it, and if they say it works well, then I will do it. So I'm not the kind of person who will try different. So I've never tried caloric restriction or rapamycin or metformin. I've never tried them myself because I think I'm relatively healthy, you know, knock on wood. I exercise. I try not to eat too much junk food. And so compared to that, I mean, compared to exercise, I think Matt Caroline was not too long ago either. And he's the one who says, you know, exercise for humans is still the benchmark. That's the gold standard. You know, you need to show something better than exercise. I'm not convinced there's anything better than exercise. So I exercise. Anything else, I wait for other people to do it. Now, I'm all in favor, or not all in favor, but I mean, I'm fine that other people like Brian Johnson are willing to try this other therapies and test it. I think that's, you know, all fine by me, and that can provide insights. I suppose as my statistical mind is going, you know, but that's just one individual. You know, you need to have a little more than one, because if you're trying everything, this was a problem actually with some of the early caloric restriction trials in humans. So people who did caloric restriction also tended to do other stuff, like take supplements, like, you know, take quite healthy lifestyles. So how do you disentangle that? How do you know that the benefits they're taking is from caloric restriction, not something else that they're doing? So that's one of the big problems, and that's why you need controlled, you know, placebo-controlled clinical trials to establish if each of these interventions is beneficial. So, you know, I like the fact that other people are trying these things and they tell me about it. I know people who take rapamycin and metformin and do caloric restriction, you know, and they tell me about it. But, you know, again, my statistical mind still goes, well, yes, but to be established as something that retards aging, you need to do like a proper study, do a statistical test in the end, and see if it works or not. Because until then, there's so many potential confounding factors that you know, that can take you the wrong direction. So you need to take that information with a grain of salt.
Yeah, it's a good point.
Does that make sense?
Totally makes sense. Yeah, it's a good point on the confounding factors. I mean, you're not going to know, is it the caloric restriction he's doing or the exercise or the specific diet? You know, which one's having the strongest influence? So definitely a good point. I find it a good proof of concept to, you know, at least measuring these different things, but I 100% agree. We need more data, bigger than n equals 1, and run some statistics on it. So thanks.
Yes. Thank you. Yes.
Wonderful. I would like to ask a little bit more about like more generic questions that we usually like try to get to in this group, which is one, like if everything goes well within let's say, you know, biopreservation, let's maybe pick that one as a focus for now. Then, you know, where do you see research progressing to in, like, let's say 5 years or so? Like, what's something where you're like, okay, now I'm feeling pretty confident that we're on a good track here. And are there any specific things you want to draw people's attention to as to, you know, what new people kind of streaming into the field could do to like help progress along in this area?
So yeah, so those are great questions. I think in cryopreservation, okay, 5 years from now, what I would like to have, I'd like to have some, say, new chemicals or new chemical combinations that, you know, improve efficiency, improve cell survival, that you can scale up with higher concentrations of cryoprotectant in a safer way, in a less toxic way. So that's what I would like to have, because that's the road not just to biostasis, but to lots of other clinical applications or medical applications of cryopreservation, because you can, you know, we have a lot of commercial value as well, and have a lot of medical value. So in terms of organ, I'm talking about things like organ preservation, stem cell preservation, and so on. So preservation of clinical specimens. So I think there's a lot of potential there to make discoveries. And so in that regard, I think having some initial findings, some initial proof of concept that, hey, you know, we can get this better, I think would really make the whole field optimistic. I think one of the things I've noticed is that, I mean, when I studied in aging, the field was quite small. I mean, it's grown a lot. It's fantastic. But cryopreservation is small. It's tiny, even compared to aging 20 years ago. So I think having some, let's call it the Sputnik moment for cryopreservation, I think that would be really, really important to catalyze more progress. And that just, I mean, there's only so much one lab or one company can do. That's the bottom line. So you really need to build an environment and build an ecosystem. And if we can make some initial findings and some can show, hey, you know, we can improve on the protocols, we can make it better. We're making advances. This can have medical applications even before we get to biostasis, you know, and that I think will energize the whole field, a bit like it happened in aging. So that's what I would like to see happen in 5 years from now. And your second question, I think, was about, sorry.
Oh yeah, and also if you, because I just realized we had time and I don't know how much time you have for us, but if you want to weave into your response also what people can do if they're particularly excited about your work in particular, that would be really wonderful.
So I think it's I mean, we do have a lot of, I mean, feel free to drop me a line if you're excited, if you want to collaborate or contribute in some way. I mean, actually, let me tell you a quick story. The initial study of allantoin and rilmenidine that we did, there was actually a student in India who contacted me because he needed to do a final year project. He was interested in bioinformatics and aging. He asked me if he could do some analysis with our lab. And I said, okay, yeah, you know, why don't we do this, drug repurposing? I mean, we haven't really done much on it. Why don't you give it a go? And I've never seen him in person. I've never met him. Maybe it's some AI. It's not a real person. It's just an AI. I'm fine with that because he gave us wonderful results that really drove a lot of research into this topic and really fostered, the findings of allantoin and rilmenidine and so on. So it really started with just an intern in India doing a small project with us. So I think if people are interested, I'm always happy to look into collaborations, partnerships, internships, you know, that there's, I think actually, so it's, I think, normal for scientists to complain that they don't have enough money. And of course, more money, more investors, I know that's great, but I think we also need more talent, you know, we need, so that's really important. And when I look at some of the very good projects that we developed in our lab, a lot of them were, you know, just interns, graduate students, some very talented, ambitious PhD students, you know, so people, you know, so not people that I hired for a job, just very motivated PhD students or graduate students that really made a difference. So I think there's a lot of ways that talented students, researchers can contribute. And of course, feel free to drop me an email.
Wonderful. Okay, that's actionable enough. Thank you so much for this nice overview of multiple different, I think, efforts that you're involved in, and then finishing it up on the biopreservation note. I think that many folks within Foresight have been signed up, at least to various different cryonics organizations since quite some time, and I definitely I think would find it really useful to have more progress occurring in that area as well. So if that's you, then perhaps you can also reach out to Joao and see if you can support them in some way. Okay, thank you so much for joining. Thanks everyone for hopping on and for your great questions. And we'll be seeing you guys, I think, in a few weeks with the next meeting, next February meeting. And until then, I would welcome all of you, for those of you who want to apply to our workshop. If you do want to kind of get a chance at just seeing what can happen at the Frontier in Longevity in April in San Francisco, if you're close. But for everyone else, I'll see you online, and thank you so much. It's so wonderful to finally have had you properly online, because your research has been circulated so much within Foresight's community. So thanks a ton also for your effort and for pushing along. Okay, thanks everyone.
Thank you.
Have a good rest of your day.
Bye-bye.